Hi, in this video, I will talk about allergy, which is otherwise known as type 1 hypersensitivity reaction. So, the hypersensitivity means your immune system is hypersensitive. The immune system generally protects our body from external pathogen attack and viral or bacterial attack. But when the immune system is hyperactive, then a situation is created, which is known as hypersensitivity. Instead of doing good to our body, it actually harms our body. And we would look at how this happens. Now, among 300 million US population, roughly about 100 million people are suffering from allergies. And the trend of increasing allergy is randomly increasing day by day. Also, in the third world countries like India, Malaysia, Vietnam, etc., the trend of getting more allergy is increasing. So people are allergic to sometimes gluten, sometimes sesame seed, eggs, sometimes even crustaceans like prawns or otherwise peanuts, etc., etc. Now, some substances which cause allergy are called allergens. For example, pollen grains. Otherwise, substances present in bee sting could also cause allergy for some people. Also, substances present in dust mite or cockroaches are also allergic for some people. Also, there are many food substances to which people are allergic. Now, some allergens are very complex and they have enzymatic activity. Mostly they have protease-like activity. Allergens found in cockroaches and bed bugs have this protease kind of activity, and they cleave the tight junctions between the epithelial cells, which is making these cells even more loosen, allowing more pathogen to enter or damages this cell or also decrease the integrity of the cell. Other proteases, such as DREP1, produced by a dust mite, cleave active complement cascades and thereby creating inflammatory responses. Now, let's say this person is inhaling a pollen grain. The pollen grain would go from its nasal pathway and it would end up in the alveoli, where in the alveoli the pollen grains would enter the epithelial cells. Now, inside the epithelial cell layer, there are several cellular players like B cells, and this epithelial cellular layer is pretty rich in IgE producing B cells. There are also patrolling macrophages and dendritic cells. Now, once a B cell encounters a pathogen, it engulfs that pathogen and display the pathogen onto the surface of onto its surface by class 2 MHC, and this is displayed to a Th2 type CD4 positive T helper cell. Now, Th2 positive CD4 helper cell would secrete interleukin 4, and that interleukin 4 signaling would allow differentiation of IgE producing B cell to a plasma cell which secretes a hell lot of antibody. And IgE is one of the important mediator of allergy. Now, type 1 hypersensitivity is solely dependent on IgE antibodies. Some people have more IgE in their blood, while all the normal people have very less titer of IgE in their serum. So IgE, more number of IgE or more Th2 cell, these kind of things are actually genetically predisposed. So this allergy could have a genetic basis also. Some people are referred to atopic are more prone to allergy. So in some families, allergy is quite prevalent to a particular substance. Now, another key player of the immune system is the mast cell. Mast cell has specific receptors for antibodies, which are known as Fc receptors. Now, Fc receptors would interact with the IgE molecules and they would become a sensitized mast cell. And the second time, when the body would encounter the same allergen, then there would be a huge reaction in the mast cells and the mast cells would degranulate. Now, once mast cell degranulate, it would do a tons of things, including smooth muscle contraction, vasodilation, bronchoconstriction, or glandular secretion. Now, what happens when for the second time the allergen is binding to a IgE receptor, molecule? In the IgE molecule is actually bound to the surface receptor on the mast cell, the Fc receptors. Now, the Fc receptors have immunoreceptor tyrosine kinase bed activation motif. Several kinases of Lin family cross-phosphorylates the ITAMs of the receptor C-terminal domain and thereby ultimately activating MAP kinase signaling pathway or PKC signaling pathway, which ultimately give rise to transcription of several players, all of which can ultimately help in production of inflammatory mediators like prostaglandin, leukotriene, histamine, or other inflammatory cytokines. Now, one of the most important mediators was histamine. Histamine is a biological amine or a modified version of an amine which would bind to the GPCR, histamine receptor. Histamine receptors are of broadly three types: histamine receptor 1R, histamine receptor 2, or histamine receptor 3. Roughly, right now, histamine receptor 4 is also discovered. All these different histamine receptor and histamine interaction would lead to different signaling cascades inside the cell. Now, all these things would ultimately lead to increased vasodilation, increased gastric secretion, increased bowel peristalsis, and also bronchoconstriction. Now, all these inflammatory mediators and all these kind of mediators which are produced by the action of interleukins, the prostaglandins, the leukotrienes, the histamines, etc., all these things would ultimately lead to smooth muscle construction. It would work on neurons. It would affect the vascular epithelial cell. It would also affect the hematopoietic cells to increase hematopoiesis. It would also work on epithelial cells. All these things lead to change its functionality. From a few hour time point to for a few days, it would evoke inflammation. Then it would generally lead to acute change in function, and ultimately it would cause in a remodeling of tissue. Now, apart from the activity of the B cell and the Th2 helper cell, another important factor is the dendritic cell. Now, dendritic cell can also engulf the allergen, here the pollen grain, and can display that onto its surface bound MHC class 2 molecule. It can go and migrate towards the lymph node, which is the base camp for T lymphocytes and the B lymphocytes. Now, it can activate the T cells there, and after T cell activation, the T cell, the T helper cell, can interact with the B cell, and the B cell would ultimately give rise to antibody producing IgE type antibody producing plasma cells, which means it would secrete even more IgE antibodies into the bloodstream, and it would work in a vicious cycle to increase the allergic responses. Now, the manifestation of the allergic responses includes redness, rashes in the skin, sometimes watering of the eyes, tearing, and sometimes running nose or increased mucus secretion, etc. Now, what is the test for allergic responses? So there is an allergic response test in which several points in the hands are marked, hands or the shoulder is marked, and then little bit suspected allergen is injected there, or the portion where you can see the marks are cut and a little bit amount of substances are there, and after about a 30 minutes or so it is checked for redness and swelling. If you find redness and swelling, that means those are the potential allergen to which the person is allergic, and this is one of the important tests for allergy. Now, treatment to allergy includes anti-inflammatory medications, and best way to avoid allergy is to avoid the food or the substances to which you are allergic. Another interesting aspect of it is using antibody as a treatment. Omalizumab is one of the antibody which binds to the Fc region of the IgE antibody. Since IgE is one of the most important mediator of allergy, if you inhibit IgE, then IgE cannot bind to the Fc receptors on the mast cell and cannot make the mast cell to degranulate and secrete all these inflammatory mediators, and thereby suppressing the allergy. In my next video, I would talk about anti-allergic medication in details and the mode of their action. So if you like my video, give it a quick thumbs up. Don't forget to like, share, and subscribe. Thank you.